Hi, I'm Darby Santiago, and I'm here to introduce you to meta-analysis. Meta-analysis is a subset of systematic review which sits at the top of the hierarchy of evidence. This short lecture seeks to differentiate meta-analysis from systematic review, describe the process of creating a meta-analysis, discuss its functions, advantages, disadvantages, and even the criticisms about this research method. Now, why do we do systematic reviews in the first place? In the past decade, there has been an exponential increase in the number of published articles in an increasing number of journals per year. This simply makes it impossible for any one individual to read them all up, much less try to sort out the worthy ones for reading from those that are of poor quality. Therefore, separating the insignificant and unsound from the salient and crucial is needed. Reviews can be narrative or systematic. They have some overlapping characteristics. Meta-analysis, on the other hand, is a subset of systematic reviews. Narrative reviews are done when a researcher wants to know more or consolidate all available knowledge on a certain topic. There is no predetermined research question, nor a specified or systematic strategy to look for and select papers to be included, and then make some conclusions in the end. Because of the convenient sampling and non-systematic way of looking through the literature, there is a chance that some articles will not be included on purpose or by chance. This introduces bias. Therefore, the saying that the absence of proof of benefit is the proof of absence of benefit may ring true. In contrast, a systematic review attempts to collate empirical evidence that fits pre-specified eligibility criteria to answer a specific research question. The key characteristics of a systematic review are: 1. A clearly stated set of objectives with predefined eligibility criteria for the study. 2. An explicit, reproducible methodology. 3. A systematic research that attempts to identify all studies that meet the eligibility criteria. 4. An assessment of the validity of the findings of the included studies. And 5. A systematic presentation and synthesis of the attributes and findings from the studies used. The goal is to minimize bias to get more reliable findings from which conclusions can be drawn and decisions made. Now, meta-analysis, as a subset of systematic review, uses the quantitative approach, wherein you get the data from all the studies and pull them all together to arrive at a specific conclusion. These conclusions may include a more precise estimate of the effect of treatment or risk factor for a disease than any individual study contributing to the pooled analysis. In other words, you are putting together in a methodological manner numbers or data from all the available studies in order to create new knowledge. By combining the samples of individual studies, we increase the sample size, thereby improving the statistical power of the analysis, as well as the precision of estimates of treatment effects. But when did this meta-analysis start, and how did it come about? In 1952, Hans Einzek declared that giving psychotherapy to mentally and psychologically unstable individuals did not help them a bit. It's nothing. So psychotherapy is useless. This started a debate because not all believed that. And that this debate lasted for more than 2 decades. And this which yielded hundreds of studies, each of which have differing results. Finally, in 1978, Jean Glass collected all the research papers involving psychotherapy. He statistically aggregated their results and came up with a conclusion that psychotherapy works and is a useful tool in the treatment of psychological disturbances.
This next slide shows us some really familiar names. No? They were the ones who gave their earliest concepts of meta-analysis. In 1904, Pearson, he averaged correlations for typhoid mortality after inoculation. And he did this across 5 samples. Fisher in 1944, he gave the source of the idea, he was the source of the idea of cumulating probability values. And in 1953, Cochrane, he discussed a method of averaging means across independent studies. This pretty much set out the statistical foundation for meta-analysis. Meta-analysis are useful in cases where the topic you are dealing with is quite complex, including the variety of literature involved. It is also useful when there are many published articles dealing with a particular topic or hypothesis, and you find that the results of these studies are contradicting, with some having positive results, the others negative, and yet some reveal neutral results. A meta-analysis can settle controversies arising from apparently conflicting studies, answer questions not posed by the individual studies, and generate new hypotheses. Some of the strengths of this method is that it imposes a certain amount of discipline on the process of summing up research findings. It represents findings in a more differentiated manner than conventional reviews. It can find relationships across studies that are otherwise obscured in other approaches. It protects against over-interpreting differences across studies. It can handle many studies without any limit. Its weaknesses is that it requires a good deal of effort on the part of the team of investigators. The mechanical aspects of this method don't lend themselves to capture more qualitative distinctions between studies. It treats the studies like apples and oranges. Their comparability is often in the eye of the beholder. It can include blemished studies. Selection bias poses a continual threat. Analysis between study differences is correlational. And last but not least, certainly not the least, there is heavy reliance on published studies leading to publication bias or the file drawer effect. This happens when studies with non-significant difference results end up in the desk drawer of the journal editors instead of being published in the public domain. Publication bias should be seriously considered when interpreting the outcome of a meta-analysis. As such, the major limitation of meta-analysis is that it can only reflect what is published and searchable. It is primarily focused on mean, effects, and differences between studies when in reality, what really matters is the effects on individuals. A third limitation is the fact that the relation between group rates and means may not really resemble the relation between individual values of exposure and outcome. Other criticisms of meta-analysis is that there could always be overgeneralization. It ignores qualitative differences between studies. But perhaps the harshest criticism is that while proponents regard meta-analysis as being objective, it is really subjective because it relies on shared subjectivity rather than objectivity. In conclusion, no single study, whether meta-analytic or not, will provide the definitive understanding of responses to treatment diagnostic tests, or risk factors influencing disease. Despite this limitation, meta-analysis remains a useful methodology for making sense out of conflicting research studies, make findings more individual studies more applicable to clinical practice, and integrate the best available evidence from research with clinical expertise to achieve optimal patient outcomes. Here are my references. Thank you for listening.